¿Listo? Bueno.
Today you have the red hot chili peppers.
Okay, well, we're going to Cantina, right? So today, we're going to talk about gene editing, right? And many of you have probably learned about something called, yes? You heard of that? Yes. Yes. And it's a good system when you have no system. But this is a much better system, what we use in bacteria. And it's called lambda red, meaning it comes from the lambda phase that we talked about. P22 has the same thing. The red is just historical. It has to do with the color phenotype on McConkey lactose plates. And it's nothing to do with anything else. It's just a historical name. Stanley likes to call these things red swaps. And there's Don Court, who works in E. coli, likes to call it recombinering. They all mean the same thing, but they're all used to do gene editing. Okay? And it changed my life. Okay, so what is it? It has to do with lambda recombination genes. So we talked about how we can maybe have, let's say, a TN10 detet insertion in His. We can grow P22 on this string, and it'll make more phage, but it also packaged the chromosome. And so we can use that to introduce that His TN10 detet by a fragment. It may be only 1 out of 1,000, but that's okay. And we can get recombination into the chromosome to replace the His chromosomal locus with our TN10 detect, right? And that's catalyzed by the host recombination system. REC-A is the enzyme that does it. But REC-A, in order to catalyze recombination, the homology it needs is on the order of thousands of base pairs. Okay? So it needs a lot of flanking DNA on either side of the marker you want to bring in for that recombination to occur. Lambda recombination system, there's 3 genes, exo, bet, gain. And the real recombinase is this guy. So this is like REC-A. That's the recombination gene within this lambda red system. Okay, so the exo part, just if you have a piece of DNA, you need to have a little bit of exonuclease to give you a single strand, even with REC-A. REC-A needs REC-BC nuclease to chew off a little bit to release a single strand. So that's the purpose of that. GAM actually inhibits the REC-BC nuclease of the cell so that only this recombination function is working. But the key guy is this, BET. And so what's so special about BET? So with BET, if we were doing the same recombination, instead of needing thousands of base pairs, we need tens. Okay? So I can do the same thing with only very small region of flanking homology. And what's so important about that, this is expensive. This you can buy. And they're called oligos. Okay, so you can make oligos. You can buy oligos, which have tens of bases of flanking homology. So, great. And what I'm going to talk about today, so we can we can do this. We could actually, remember TN, 10D, TET, looks like this. There's TET R, TET A. And it could be anything you want. Let's say you want to bring in you have a piece of DNA that's chloramphenicol resistant, myosin resistance, GFP, doesn't matter. Okay. You can take any piece of DNA and you can amplify it by PCR and target it anywhere in the chromosome. So if we target, let's say this I call tetra. It's easier to draw than all that. So I like to use that for my tetracycline resistance element. And chloramphenicol, kanamycin, GFP, whatever you want. Okay. So let's say we have a piece of DNA that has this tetra element. And instead of doing a hop and finding these insertions, I just want to make it. I just want to make it. And I want to put it in a specific place. I want to put it in a particular his gene, his D. Okay, how do I do that? So I need a primer, oligo. Maybe have 18 bases to amplify that tetra, and then 42 bases of his D. Now, the reason you don't have to do this anymore like that, but the reason historically we've always done 42 for our recombination and 18 for our PCR is because when you buy oligos from IDT, after 60 bases it becomes very expensive. So, now it's not the same, but that's just historical. So I can have a particular base in his D, I'll call it, let's say codon 161, 162, and so here's 161 right here, and then earlier codons. And now I can have a primer on the other side, starting with codon 162, 42 bases, 18 bases. And when I PCR amplify that, what do I get? I get a tetra element, or it could be GFP, kanamycin, chloramphenicol, whatever you want. And now I have 42 bases of his D. Okay. And then starting 161 is the end before the tetra. 162 over here. That is my DNA fragment that I made by PCR. Right? Pretty simple. Now I can electroporate that. Into a recipient, and there is several plasmids. Barry Warner created a plasmid called PKD46. We use something called PSEM5. There's a whole lot of plasmids out there that have some selectable marker. Could be, I can make this AMP. If I want to use AMP, I'd make this chlor. And it has the arabinose promoter driving the exo that AMP. So if I electroporate this into cells, in the presence of arabinose, and this plasmid, or PSEM5 is the same thing, but it's under the control of lambda repressor, TS. So lambda promoter driving exo bet GAN. Okay. Fabian, my research professor, likes this one better. Traditionally, this was the only one available. But anyway, anything, so this would be induced by heat. This would be induced by arabinose.
But does she like it better? Because I remember in the beginning of this technology, the PKD46 was kind of finicky, like sometimes worked, sometimes it didn't.
Arabinose can do things to the osmolality of the cells. It can make things, a little finicky.
So
It's called empirical, or empiricism.
mm-hmm.
We try this, it works better, we use it.
Okay, good.
But sometimes you might be dealing with something where you can't do a temperature sensitive
Yeah.
shift, right?
Okay.
So then you can go back to here.
Okay.
So now we bring in this fragment. And we allow the lambda red recombination functions to bring this in. And now we insert that tetra between codon 61 and 62 of his D, exactly where we targeted. We could have targeted anywhere in the chromosome just by what's flanking that. As long as we have a selectable marker, in this case the selectable marker is tetracycline resistance. Okay? So, we like this tetra element, but you can use other elements for a process called selection, counter selection. So one of the beauties of the tetra element is that you can select for it. Tetracycline resistance. You can also select against it, because if you have the TET-A pump in your membrane, you're fusaric acid sensitive. So you can counter select against this by plating them on plates with fusaric acid, and they'll die. So, what if what I really want is a deletion of the his D gene? So I have in my chromosome now, so I'm going to take it out, draw it just there. Here's the his D. Here's his G. GDC. And I want a clean deletion of that his D gene. I can simply buy an oligo. I gotta buy an oligo to the end of his G. And then an oligo, that oligo has the beginning of his C. So I buy that. I buy that fused together. Okay, G, C. I introduce this by, I fill this in. I can make this as long as I want. The longer, the more recombination. And in the presence of lambda red, so the plasmid, either an era-induced lambda red or a temperature-induced lambda red, I can plate these on fusaric acid plates and look for fusaric acid resistance. That allows me to select for those guys that replace the tetra with that piece of DNA, and now I have a clean deletion of the his D gene. And I could leave as much as I want. So normally you don't want to take out the entire gene, because at the end of a gene before the next is the ribosome binding site for the next gene. Right? So I typically leave 15 amino acids, to ensure that I have the ribosome binding site for the next gene, just to make sure. Pretty nifty. And it's clean. The FDA will say that's okay, there's no drugs left over. Right? Now we can use it. Right? We can inject it in humans for vaccines.
You have to keep the frame when you do the deletion.
You don't have to keep the frame, but if you want normal expression of the downstream genes, I like to have, usually just leave 30 amino acids. So that ribosome binding site, which is evolved for the his D gene, right, still there, and the ribosome binding site that's evolved for the his C gene is still there. Otherwise, I'd have the you know, this ribosome binding site, which is potentially artificial for that gene. I like to keep things as natural as possible. Okay, now what do you want to do? Give me a problem. What do you want to do?
Using the same technology, I'm not sure who developed it, but adding tags to the genes.
Adding tags to the
Yeah,
genes.
like the Yusau, was that Nello and Nara?
You want to tag? Well, let's tag it with GFP. That's a pretty good tag,
Okay.
right? How would we do that? Okay. I just told you that we can target that tetra to any gene, right? I showed you how we can do that. So that means I can put, let's say we stay in the same gene. Well, let's make it your favorite gene. Your favorite gene. We can put a tetra at the very end. Right? And then we can come in and PCR an oligo that has the end of your favorite gene. And then we have a source of DNA that has GFP. Right? Here's our 18. This side, 18. And then whatever this is, let's call it XYZ. XYZ. And now we have a fragment that has your favorite gene. And then before the stop codon, we put in a tetra, so now we can just have GFP. So that we have an in-frame fusion, and then XYZ. And now again, on fusaric acid. So tetra, putting it in target where we want to go, and fusaric acid resistance to counter select and replace that tetra so that our product is now your favorite gene that's fused to GFP. Okay? Okay. I'm going to do just a little fun experiment that we did with this. And you can ask for whatever you want, we'll make it. Right? I mean, this won't make your coffee in the morning, but it'll do everything you want in the lab. All right, let's do something fun. And think about it. Think what you want. Because this thing will do anything. You can target anywhere. This way, it is really awesome. Okay. We're going to look at one of our favorite genes. Remember our flagellum gene? Remember that guy? Okay. All right. We talked about how in that gene we have mud J's, mud K's. This is a transcriptional fusion. This is a translational fusion. I want to look at the promoter region for mutants that are defective in transcription and those that are defective in translation. And I'm going to use lambda red. How am I going to do that? Well, here's the promoter region. There's a 5' UTR. There's an ATG. And then they're coding. Okay. I can PCR a tetra up that'll replace this whole early region of coding with a tetra. That's deleted. So it starts here, and we put in a tetra. So we target a tetra that deletes this whole region. Okay? And we can come back and just from a wild type DNA chromosome, we can use PCR primers to amplify the promoter 5' UTR, and then the first coding, the N-terminus, we can PCR this region up, CR, under mutagenic conditions. Okay, so we can, there's ways of doing PCR that make mutations. Okay. And you can tune your frequency mutations to try to get only one hit per amplification. Okay, so far so good. So now we can take that fragment, let's draw it over here, all right, with some mutation, and in the presence of the lambda red, so P if the plasmid's there, era or temperature sensitive, on fusaric acid plates. We usually call this tent sensitive plates. We replace that tetra fragment with the region that has the promoter, the 5' UTR, and then the starting N-terminus. And we get colonies. Okay, these are tent-sensitive colonies. Okay. Now I can check these on motility plates. So on a motility plate is a soft auger plate, and you can inoculate in the center. It has tryptone, and there's two favorite sources of carbon. One is serine, and one is aspartate. That bacteria like E. coli and salmonella will chemotax to. So when you inoculate with a toothpick from a single colony, they'll immediately start eating the serine and aspartate, and by consuming it, they set up a concentration gradient. Pretty soon it goes to 0, and they start swimming. And then you wait about 4 hours, and you'll see these rings as they swim out. This is denser cell growth. This is a serine, your favorite. And the ones get left behind say, darn, no more serine, but they'll swim to aspartate. You'll see these chemotaxis rings. And I'll show you that tomorrow. So tomorrow you're gonna get a little treat. You actually will get a slideshow. Not
Hey, Kelly,
Yes.
it'd be useful for people to have an idea of how many you would have to screen through like that to find what you want.
So because you targeted a very small region of the chromosome, this is like 100 bases, right? And if you set up your mutagenesis right, you can have it so that maybe 1 out of 10 has a phenotype. So we can pick those on motility plates, and we look later. And here's two guys that don't swim. Okay, so they're mites. So now, we have some mutation in the promoter 5' UTR, in ATG, or the region that we targeted. How are we going to know if they affect transcription or translation? Well, we have our reporters. So we already have this delta promoter UTR tetra. We can just transduce that in here. So upstream we have so we can grow phage on this, our donor guy, and move it into our muds to bring in the tetra and keep kanamycin. Same thing with this. Okay. We can make those fragments just by P22 transduction. And now we can go into this strain by transduction. And what do we do? So if our donor is delta tetra with a tetra insert, and then downstream we have, let's see, a mud J. We select for kanamycin, and we screen for TET sensitive. That means we brought in the mud J, but we didn't bring in the tetra. So we brought in the mud J, but we kept whatever our mutation is. So now we have some mutation. And it could be lac minus or lac plus. Right? If it's lac minus, and this is a transcriptional fusion, we have a promoter mutation. Okay? But if it's lac plus, we did the same cross with that mutation. It's lac plus with a mud J, but now with a mud K, we bring that in, the lac plus is now lac minus. That would be a translation mutant. Okay. And if we brought in, was lac plus here and lac plus here, but it was mode minus, it was in the coding sequence. So it was making a mutant gene product. Okay. So we can, using these MUDs, we can differentiate whether we have a promoter mutant, translation mutant, or the mutation in the N-terminus of the protein. Okay. And so let's look. Let's look. So fly C, P, fly C, minus 35, minus 10, plus 1. Then we have the 5' UTR. And if we look at that 5' UTR, it forms some stem loop structures. We get to the ATG. And if we look at our guys that are defective in translation, so if we put in, if we have the mud J, that's lac minus, what's going to happen if we put in mud K? It's also lac minus. If you don't transcribe, there's nothing to translate.
Yeah, sure.
Okay, so those will be
Lac
minus in both. It's only the translational guys that will be plus and then minus. So the ones that are transcription, guess what? They're all, here there's a deletion. They're all in the minus 10 and minus 35. Yay! What about the translation guys? Green. They are in these two stem loops. And usually at the very 5' end of a transcript, the stem loop is protecting it from degradation. So this usually involves stability. And this was a weirdo. There was a, let's see, it's CCCG, that's right, GGGC.
Make sure you get that in your notes.
It's
microprint.
All right. We had a C to U change. A C to U change. And that just, instead of a GC pair, it was a GU pair. So instead of 3, it's now just 1. And just to make a long story short, without telling you too many details, that actually causes this stem loop to open up instead of being closed all the time. And what we found was that in the coding region, downstream, this stem loop 2 sequence could actually perfectly pair with some of the coding sequence to inhibit translation. Why? I have no idea. We have to figure that out. But it gives us something to go study. Right? Okay, yeah.
Kelly, wouldn't you have expected to find mutations in the ATG also?
Oh yeah, we did. Sorry.
Okay.
I'm sorry. Yes, thank you, Stanley.
Could you
Yes.
comment on frequency of the two types?
And ribosome binding site.
Yeah.
Sorry.
That's what I thought too.
Yes, 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 yes. Thank you.
Could you say something about frequencies, just
Frequencies of.
of stem loop, ribosome binding site, ATG?
Stem loop. No, they're
About the
roughly
same?
about the same because they're all small targets. Yeah, 3 codon, 3 bases, 6
Right.
bases.
But I would have expected that there's a lot more ways to screw up a stem loop than to screw up the small sites.
Yeah,
Yeah.
it's about the same. It's a small stem loop. Yeah. Okay. So I wanted to give you some example of how that can be used to do whatever it is that you want. So tell me what you want. We can do it.
Yeah, in my case, I was thinking we can do using this tool, especially with the tetracycline, we can generate also mutagenesis in our favorite genes,
Yes.
which is CBEF and CK
Yes.
for now,
Yes.
and then substitute that directly into the chromosome, and then we'll get results because we already have something to test. In this case, sub-B expression. That's what we test. But really, let's see.
But you can imagine that you have in your gene, with your favorite gene, codon 161, which might be a serine, right, a UCA, and you would like to change that to a tryptophan, UGG. You simply target your sequence to delete it and replace it with an oligo that now has UGG, right? Or you just wanted to see how important that serine was. You come in with an oligo that's just N and N. And you ask which ones work, which ones don't work. I mean, you can go on and on and on.
In
You
that
can do
case,
anything.
that oligo needs to have 18 bases.
No, we would buy an oligo that had at least 40,
40, okay,
but
20, something for each.
the
for each
longer
side.
the flanking sequence,
The best.
the frequency of recombination goes up exponentially.
Oh, okay.
Okay.
So what is the minimum for that, 40? 40?
Actually, the minimum is, it's published, it's more like 22,
For
but it's very low
recombination?
frequency.
Okay.
And as you go up, it goes up exponentially. Like I say, we did 42 just because of the price of oligos. After we got past 42, it got more expensive, but now it's not that way. So the longer, the more homology,
And it can be used for SNPs?
it can be used for anything. What do you, a SNP, I just made a SNP. I went from serine to tryptophan, right? That's a SNP. Or I could have made a single base, a serine codon to a different serine codon. A SNP, a synonymous mutation, right?
And
Yeah.
you could put 2 fragments at the same time, or it's just like?
I first put in the tetra, and then I
Yeah, but
replaced it
if
with an
I
poly.
want to put, I don't know, tetra and GFP, I could put in the same time at the same,
You want to do it one at a time.
okay.
So once you do this, and you remove the tetra, when you replace the tetra, you can now put in another tetra. You can do it again, and again, and again. I mean, remember sigma 28? Those class 3 promoters? There's 17 of those. And we went in and deleted all 17, one at a time. It took a while, a few months, but we made a strain with all 17 promoters deleted. Yeah.
That's really cool.
And all you need is selection, counter selection. So other people use things like, There's something called SAC-B, fused to kanamycin. This is the selection. This makes you sucrose sensitive. So you can select against it on sucrose plates. So whatever you can use that has a selection, counter selection will work. I talked about that ATSR in Burkholderia where we had a T23 insertion. Well, we went back and did a lambda red to delete that. Right?
Can you use lambda red?
And then we could do the T23 transposon mutations in that background to knock out the antibiotic genes, right? That's what we did.
So the same system worked for you in Burkholderia? The lambda red?
It worked for
For
us.
Thailandensis.
It doesn't work as well as salmonella because the coding of Burkholderia is different than, but it worked fine. We got it. Chris got it.
Ah.
Yeah, I have a friend, colleague that works in these bacterial symbionts. They're called Sedalis. Lambda red works great.
Huh.
So there are systems where that promoter might not be expressed in whatever that particular bacterium, in which case you'd have to engineer your own expression. Okay.
Good. Pretty awesome.
Ready? What should I talk about CRISPR? You guys know about CRISPR? We talk about more red swaps? I hate CRISPR. It sucks. In bacteria. When you're used to this, I mean, if you're working on fruit flies where it's really hard, it makes sense to do something that's really hard like CRISPR. But if you have something like this, it's so easy, right? Why would you change? Did you have a question?
Yes. Is there any limit for a gene to insert, or you can insert any?
Right. So there is a limit. And that's one reason why we would use CRISPR. So, for example, there is a tRNA gene we work with, I'll talk about tomorrow, and it's called serine T. Serine T is an essential tRNA. And you see, around base pairs 25, around base pairs 42 of that tRNA, not base pairs, sorry. Well, I guess if you think about the DNA base pairs is okay. We would like to target the mutagenesis of that. It's an essential gene, we can't put a tetra there. Right? But we can complement that. We have to have a plasmid that's serum T plus, and then we can complement. But we could also use CRISPR. CRISPR allows you to do gene editing on a cell, on a wild type gene, right? So that's one advantage of using CRISPR. If you can't do a complementation like this. Coffee time, unless you want me to make another one. Your favorite gene. Yeah.
Come on.
Maybe we could insert recombinant gene, maybe to make like a spider silk-like protein.
Spider silk is really hard to express. I've tried it. It's all proteins and glutamines and glycines. It's just a mess.
So complex.
Yeah.
But maybe we could replace non-essential genes with all of the proteins needed to form the cell.
Yes, absolutely. Absolutely, yes.
Let's see, we find some genes that are very good, that are in the synthesis route to make an antibiotic. But the promoters are really poor. So, then it makes really few, a little bit of that antibiotic. So then we need to improve the promoter, something like that. Do you think? Change the poor to strong.
Sure, you can replace this with a tetra, and then you could replace it with a strong promoter, or an inducible promoter, whatever you want. You can put
Mhm.
the lac promoter and then right upstream lac I to control it.
Mhm.
Exactly.
Sounds good.
Yeah.
Ah.
You can have a reporter, like, let's say you want to look at, you know, I talked about the flagella class 1, class 2, class 3. We can have a YFP and then a tetra that we put in the chromosomes just randomly, and then a CFP with a tetra. And then now we can replace this with, let's say, a class 3 promoter, class 2 promoter, vice versa. So we can look at expression of our promoters without even screwing up the flagellar system,
Cool.
right?
Nice.
Anything you want. You just have to think about it. This is a dream come true.
Yeah.
It really is. Anything you want.
For example, if you wanted to like have I don't know, an antibiotic expression as a constitutive gen, would you swap it for something that has a constitutive like promoter? Or you just like PCR it with a mutagen until you would eventually get something that
I
expresses
mean, you
it
could,
every day?
if you have, you could PCR target this to look for expression perhaps, to try to get a better promoter by point mutation,
Just replace.
or just replace it. Doesn't have, without lac I, it's a constitutive promoter. Yeah.
If I could make a comment, there are lots and lots of gene products in the cell that if you express at high constitutive levels, it's toxic for the cell and you'll get a second mutation. If you put it under the inducible promoter that Kelly talked about, now you can do a terminal induction. Induce it when you want it induced,
Yeah.
and you don't have to worry about other messing up the cells.
Correct. Yeah.
Antibiotics fall into that category of toxicity at high levels, constitutive.
Exciting.
Coffee?
Okay, can I say something? Can I say something here? There's this old saying, right? The chromosome.
Say it, Stan, say it.
Well, I was gonna let you say it, the chromosome mechanic. The pencil is the wrench of a chromosome mechanic, right? So Kelly just drew a bunch of stuff for you. He's done all those things in the lab, but before you go to the lab, you want to draw it out on paper so you know what's going on. Just like Kelly drew up here on the board, right? If you go in the lab before you've drawn all the possibilities, there's a good chance you're going to screw up, right? So Kelly is a great example of a premier chromosome mechanic, right?
Yes.
So pay attention to the pencil part.
Yeah. Good.
Okay.
Yeah, so if you're working in SPY 1, you want to delete MG, SIT, BCD, all in the same strain. You can just land and read them out one at a time.
As a matter of fact, another example is Lee Notler. She removed the whole SPY1 by using lambda ret. So it's 40 kilobases.
Right,
So he removed the whole region,
right.
SPY1 and SPY2, which is two regions that are really big. So you can delete very big portions of the genome just by using this one if you don't want to.
Yeah, so that's because let's say 30 kilobase pairs. ABC, XYZ. A DNA insertion, we can think of it as insertion, behaves like a point. So when you bring in your oligo, recombination treats it like a point mutation. It's harder the other way around. It's harder to bring in 30 keys.
Yeah.
All right?
Okay.
Cool. We love mandarin.
Vamos a café. ¿Me ayudan a pausarlo, por favor? O digo, ponerle stop. Y ahorita si me lo pasas para revisarlo. Okay, Dr. Maloy, all yours.
All right. I wanted to start with a little story that I think is really important to think about. And those of you in the lab, in particular, may relate to this. I had a good friend who passed away. His name was Ron Taylor. And he studied Vibrio cholera, right? So with Vibrio cholera, I mentioned the other day that the cholera toxin is the agent of disease. But he identified these toxin co-regulated pili, right? He isolated mutants in them. And once he isolated the mutants, he wanted to have a way of finding mutants that either lost that pili or had the pili. And what he discovered was if you took the bacteria that are TCP+, and you put them in the right kind of media, the pili aggregated and the bacteria all clumped at the bottom. Right? So positive, they clump. If there's a mutant, no clumping. Right? So it's a different kind of assay, but it was an assay that worked really well. All right? We taught together at Cold Spring Harbor Laboratory, and that's where he learned to do this, and it worked so well. Isolated a lot of mutants. He moved back to his laboratory at Dartmouth. It didn't work. It didn't work at all. He was doing the exact same thing. He was the one who did the experiments. And the difference was the water. All right? It was the water. And this is something you should always be aware of. When you're making some media, and if it's not working quite as well as other people have reported, it's always a good idea to think about the water and try different water, right? Kelly mentioned fusaric acid. Fusaric acid is a chelator. That means that if certain things are in the water, that fusaric acid is going to be in a form that it's no longer active, and the TET sensitivity will not work, right? So when you have a problem like that, that's where you just want to go back to the drawing board and say, which one of the components might it be? Make sense? Sure. All right. Just a little warning story. All right. This is what we're going to do in the next little bit. I want to talk to you a little bit about genome organization and how evolution, in particular niche, how evolution of adaptive functions can happen, right? So we're going to be talking about mutations that happen in nature, and we want to understand how they happen, instead of what Kelly just talked about, was just constructing your own favorite mutation. Right? So we're going to begin. I'm going to talk to you a little bit about chromosome rearrangements. Then we're going to talk some about polymorphisms and SNPs, and how you can study these. The particular class I'm interested in are pseudogenes, that is, a single nucleotide change that inactivates an enzyme. We'll talk very briefly about gene amplification and the fitness cost in compensatory mutations. This is from work on antibiotic resistance. And I'm going to briefly mention environmental gene pools, but we won't have time to talk about all of that. Okay? This is where we ended yesterday.
Yeah.
Right? So, I think I asked a few questions. One of the questions was where, comparing typhimurium and typhi. One of the questions was why might we see a difference like this? And one of the questions was, how can you even see that? How would you detect that? Right? So let's just start. So remember, here's the take-home point. Typhimurium strains from all over the world look like this, where these are the ribosomal RNA positions in the cell. This is the origin of replication, and this is the terminus of replication. Typhimurium, wherever you get it, it's going to look like this. Okay? This is typhi ty2, a strain of Salmonella typhi. And in this case, look, so here it's A, B, got that wrong. One too many. A, we're just naming these fragments, artificially naming them. A, B, C, D, E, F, G. Okay. Look at this. A, G, C, now E, F, B, D. So the order is all switched up. Right? Now, when you look at typhi in nature, this is just one type. There's all kinds of different types. Any type you can imagine, you can find. Right? So, what do you think? What's the difference between these two organisms? Typhi versus typhimurium.
Typhi is restricted host human.
What about typhimurium?
Subrotor.
Yeah. So typhimurium can grow inside of insects, it can grow inside of mice, it can grow in rabbits, it can grow in deer, it can grow in humans, and it can even grow in plants. Right? So, what do you think that means? What's the big defining thing about broad versus narrow host specificity?
Metabolic ability?
Metabolic ability. It might be because this has certain metabolic requirements that are only in humans, right? And this one doesn't have those requirements, so that's the thing. But let's, absolutely, there could be some things like that. What do you think it means about, let's just say, numbers of the organisms in the world?
I didn't get.
Numbers. How many Salmonella typhi do you think there might be in the world?
Oh, okay. That's a question. Only a few?
It probably less than the human population,
Yeah.
right? Because we don't all have it. What about typhimurium?
Widely distributed.
How many insects are there in the world? We have an insect expert here. Okay? How many insects are there? How many mice are there? The mouse population is much greater than the human population. Right? Yeah? So there are lots and lots of typhimurium hanging around the world. Okay? There are very few humans. Right? So if you think about what this means, there's a core genetic concept. And that concept is a bottleneck. Right? So if you start from this infected person, they may be able to infect some people in their small group, right? But then ultimately, it's probably a very small subset of people that are going to infect other people. Right? I know, I shouldn't have them standing up. Right? So that's a bottleneck. So when you think about genetics, what that means is if there's any change in here, when it moves through that bottleneck, that genetic change is fixed. Right? Whereas if you have a population where you have a variety of different hosts, right? You never have to go through a bottleneck. Every one of these has a high probability of infecting something else, right? So if a rare mutation happens and you go through a bottleneck, that rare mutation is fixed. If you don't go through a bottleneck, rare mutations often cause a little bit of less ability to survive, and so it won't survive, and the ones that haven't got the mutation will survive. Yeah? Anyway, this is theory, right? That's theory, but this is an old genetic theory that deals with all organisms that clearly applies here, right? So that's what I wanted you to think about here. And that ties to both the questions, where do they live and why the difference? Right? How could you see something like this in vivo? So the guys who discovered this, there's a guy who ran the Salmonella typhimurium Genetic Stock Center for geneticists like us to share strains. Right? So he looked at Salmonella typhimurium, lots and lots of strains for a long time. But at some point they said, well, we'd like to go from the mutations to understanding the chromosome. Right? So they used a trick called ICEU1 mapping with pulse field gel electrophoresis. Do all of you know about pulse field gel electrophoresis? Yeah? Right. Okay, so if you don't know, pulse field gel electrophoresis is simply a way of separating very, very large DNA molecules, right? Because instead of a continuous electrical field, you get a pulsed field. So you go little this way, little back, little this way, little back, and ultimately you can separate very large molecules, right? ICEU1 is special because ICEU1 cuts only in ribosomal RNAs, right? So it will cut this into 7 fragments. It will also cut this into 7 fragments, right? But when they did this, they did a partial digest with ICEU1. So from the partial digest, you can tell is C next to G or is F next to G, right? So by doing that, they could distinguish these bacteria. But holy smokes, it's a lot of work. I mean, you're not going to be able to look at individual organisms in vivo, right? Right? So you can learn about the molecular structure, but you can't really even begin to think about doing genetics, right? So here was a really interesting thing that we hadn't seen before, and it might tell us about some fundamental concepts in genetics, but we didn't have a very good way of studying it, right? So, so we wanted to see what's going on. The idea of how it can happen by recombination between these repeats, that was straight. Oh, did I tell you yesterday, each one of these RRN repeats is 6.5 kilobases? Okay, pretty big. All right, they're 6.5 kilobases. They're 99.5% identical, right? So these are hotspots for recombination, right? And that's why all of that funny stuff I said, where I showed the duplications, I showed the levitation, I showed inversions, all of that can happen because these are such good places for genetic recombination. Okay? All right, but we were really interested in understanding something about the broad genetics of this, and so we wanted to ask these questions of why are some of these more plastic than others? Plastic means flexible, right? So we had some predictions. So if one thing you might say is there might be a physical difference. Maybe this doesn't have anything to do with environment. Maybe there is some core difference between these chromosomes that restricts it, right? So if that was true, the predictions are if you looked in vivo, and in this case, for example, a physical thing might be replicor balance. So replicor balance is from the origin of replication to the terminus. How long does it take to make this half versus how long does it take to make that half, right? So if that balance was different in Typhimurium than in Typhi, it might cause constraints on Typhi, right? So that was one possibility. Another possibility was that there's a biochemical balance, a difference. Maybe this has an enzyme that promotes the recombination, or this one doesn't have that enzyme and this one does. Okay, so that's a possibility. And a third thing is that there's a difference in the ecology. So the ecology difference is what we just talked about, right? Different hosts, that's how you live in the environment. So these are three possible reasons that you might see these things happening, right? So let's take a look and think about how you can think about this. We didn't want to do the ICEU1 because it's too much work. We can't get genetics. I mean, we can look at maybe tens of chromosomes, but we can't look at hundreds of chromosomes. So a student in my lab said, oh, I'll be really smart. I'm going to use this molecular approach. I'm going to do PCR, and I'm going to build a primer set that PCR is up this, and a different primer set that PCR is up this, right? For each one of these, right? Now you can run PCR on the chromosomes, and you get discrete bands. So if this is G hooked to A, these sets of primers will work, but since G is not hooked to A here, it won't work here, right? But in this case, these primers would work. So if you do all of the primer sets, PCR reactions pretty easy. You should be able to really look at a lot of different cells, right? Right. Here's the problem. Here is our big ribosomal RNA, right? So we're going to PCR up this guy. And this is going to, let's just say this is primer B and primer A, right? And now in that same cell, we're PCRing up with primer G and primer C. So they'll give us discrete bands. But once they've generated a few bands, now this will hybridize with this, right? Because these are the same. So the PCR will begin to work really well, but as it gets going, instead of having discrete bands, we get all these big ghost bands, right? Make sense? Another biochemical technique that doesn't work, right? Bummer, right? So we needed to think of something that would be really specific. And what we really wanted was to have a genetic test to distinguish these situations, right? And especially a genetic test where we could do selections in vivo, right? To do this, we built an RRN inversion assay that used the red swap approach that Kelly described. Right? So for example, let's just say here is a sequence in the chromosome, right? I'm going to call this A, B, C, D, E. And I'll just call this RRN1 and RRN2. Right? So what we want to ask is, can we get recombination between these RRNs? This isn't much sequence, but these can be very, very far apart on the chromosome. Right? So what we did was using red swaps exactly at the end of that RRN, exactly at the end, we inserted chloramphenicol resistance. Okay? Then exactly at the other end of this one, we insert kanamycin resistance. Okay? Chloramphenicol resistance is a long way from kanamycin resistance. Right? So if we wanted to transduce from here, we can transduce chloramphenicol resistance, and it will never be kanamycin resistant. Or we can transduce kanamycin resistance, and it will never be chloramphenicol resistant. Right? Let's just say we get one of these inversions. Now, what happens? Okay, so we're gonna look for an inversion that looks like this. It comes down here. It picks up chloramphenicol resistance. It comes down here, comes back around and down. Now, in this recombined guy, we're going to have chloramphenicol resistance and kanamycin resistance, 6.5 kilobases from each other, pretty close. And down here, there's no resistance, but if you look at the outside genes, we now have, here's A, this is going to be D, C, B, E. Yeah? Okay, so now we've moved these two together. So if a recombination happened between those guys, all we have to do is grow bacteriophage P22 and see do we get co-transduction. N resistant, CAM resistant. Right? All right. Hey, you know, we can grow P22 on hundreds of cultures. We can plate them on hundreds of plates. That's really easy, right? So now we have an approach that we can test many, many samples. Even if this is a rare event in a population, we can easily detect it, right? So remember, we're probing a rearrangement that happens in nature. We're not just trying to construct something to learn about it. No, we're trying to learn about the frequency of it. We know it happens, we're trying to learn about the frequency. All right? So now that we have this test to do that, we can ask the question of how often do we get an inversion like this? So we call this an inversion index, right? So here's a strain, typhi. So these are two typhis. I'm going to just call them typhi number 1, typhi number 2, and typhimurium. Right? So the inversion index is the number that have inverted versus the ones that haven't inverted. So if you look at this, typhi number 1 is 8 times 10 to the minus 2. Typhi number 2 is 2 times 10 to the minus 2. And typhimurium is 2 times 10 to the minus 2. They're all pretty similar. Right? What do you think that means? How could you explain the fact? How could you explain the fact that typhi and typhimurium undergo these recombinations at the same frequency, but we see changes in typhi and not in typhimurium.
Just a comment. One would expect that typhi, since it has only a host, it wouldn't need to have so many changes to adapt, contrary to typhimurium, which faces many different hosts.
This tells us that it's probably not a difference between the chromosomes per se, or the enzymes that catalyze the reaction. That is, it's not a physical difference, it's an ecological difference. It's a lifestyle difference, right? It has to do with what we talked about here, potentially. It has to do with this business of the number of hosts available, right? But, you know, it wasn't something that you could do before you could do these kinds of experiments, right? By the way, I'm not going to talk about it, but this makes some predictions that if you look at people who are chronically infected with typhi, there are some people, especially in Eastern Germany, antibiotics were not prevalent when the Soviet Union started, so people would get these infections and they would just have infections. But some physicians collected samples for over 40 years. This was, for whatever reason, it was primarily women, and they have samples where they've sampled them once a year, every year for 40 years. Each one of those samples they kept in a tube. Once they put it in the tube, it stayed the same, it didn't change. But if you look from year 1 to year 2, within that person, we see these rearrangements, right? So it's something about being inside a human in that privileged situation where they are most likely in the gallbladder that this is happening, right? Look. This isn't a technique you're all going to use, but the point is, if you think really hard, you can design an approach to look for even rare things that happen in nature, right? And things like this would work if you wanted to understand organisms out in a pond or organisms in the field, or you name it, okay? All right, there we go. A niche adaptive function, we got that down. Now, what about this business of SNPs? So Salmonella typhi and Salmonella typhimurium have another difference. It's very interesting. And we didn't even know about until the chromosomes were sequenced. Right? And that is, if you look at pseudogenes, that is, genes that by DNA sequence have a point mutation that would inactivate the gene. There are about 5 times as many in typhi as there are in typhimurium. That's kind of interesting, huh? Maybe that has something to do with the difference in chromosomal arrangements, right? So, oh, by the way, did any of you read the discussion paper for today?
Yes.
Yeah? How many read the discussion paper for today? The discussion paper for today basically deals with this, and it takes it to the next level of understanding. So it's really very cool. It's by Dr. Hughes' lab in Sweden, and it's a cool paper. We'll get to that. Okay, these mutations, there's so many of them. The vast majority of them are in this part of the chromosome, and I don't know, there's over 100 of them. A lot of them, right? So now you can say, okay, maybe those pseudogenes played an important role in restricting typhi to only being able to live in that one niche. Earlier somebody said, well, maybe there's metabolic differences in these guys. Well, maybe those metabolic differences are because of all of these pseudogenes that inactivate core genes, right? Okay, so so the DNA sequencing guy said, oh, yeah, this proves that that's the difference. Does it prove there's a difference? I don't think so. Right? I think you need to test the biology. So again, we wanted to use a way of testing this. We didn't have a way of replacing a lot of these genes because some of them, the functions weren't very easy to look for. So we used a red swap approach, like what Kelly talked about. You can see we both are very fond of red swaps. Right? It's a backwards P. All right. So here's basically what we did. These are in different strains than I did before, but what we did was we had a little fragment that had chloramphenicol resistance and it had TET resistance. Right? So we could PCR these little fragments where the PCR at the end of the fragment flanked one or more of these cryptic genes. Okay? So now we can take that fragment and we can bring it in to the chromosome. And here is our one of our pseudogenes. Okay? We replace that pseudogene by demanding that it becomes resistant to chloramphenicol. Okay? Now we take from Salmonella typhimurium that carries the wild type copy of that pseudogene, right? And we bring that into our strain all right. Let me draw another step here. I want to make sure this is clear. So now what we have in this case is we have Salmonella typhi, and where there once was a pseudogene, we have chloramphenicol resistance, TET resistance, right? So that region's removed. Now we take from Salmonella typhimurium and we transduce, we select for TET sensitivity. We're getting a lot of this TET sensitive selection today, but what it is, what's left behind is Salmonella typhi with a small region of the chromosome that carries the wild type version of the pseudogene. Yeah? Got it? Make sense? Okay. So basically, if that wild type copy from typhimurium is important for some particular niche specific process, when we replace it in typhi, we should be able to see a difference. Correct? All right. So, to answer that question, we do experiments in mice. If it restricts niche, it should restrict your ability to survive in mice. And typhi doesn't normally survive in mice. So what we do is we do IP infections in mice. We're injecting the bacteria into the peritoneum of the mice. We do this in a way that we have 1-to-1 mutant, another pseudogene, to wild type. Okay? So if we start with 1-to-1, if this has no impact on fitness, then when we recover the bacteria after infection, It should be 1-to-1. Yeah? If there's no impact on growth of the bacteria and inside of the mouse, we should get the same thing out that we put in. Right? If it is important, if the pseudogene is important, then what we should find is that we should have more of our repaired wild type guys than the mutant form. Okay? This means that the pseudogene is important for that particular assay, the assay growth inside of the mouse. So let's just look at a couple. So now we're going to be looking at the ratios of these two, right? So the SOP-D gene is an important gene for Salmonella typhimurium. In this case, we look at the SOP-D knockout mutant, right? We can compare that with the SOP-D with the pseudogene. And what we see is that both of these have a minor effect. Okay? A small effect, but this pseudogene isn't partially functional. It's no better than the complete knockout, right? But it has a little bit of an effect on growth inside the mice. So we do another one, SSEJ. So we look at SSEJ, so we start SSEJ, another gene involved in Salmonella pathogenesis. We always compare with the complete knockout mutant, so we know what we're looking at. SSEJ with the pseudogene. And what we get is 0.4 and 0.4. Again, a small difference. It looks like the two are the same. So why don't we try this with the SSEJ pseudogene plus SOP-D pseudogene. Okay? Because there's a lot of them in the same time in these guys. Now, it's less than 0.1. Right? So these pseudogenes that have accumulated in typhi prevent typhi from being able to cause infection in a mouse. Yeah? The reason that might happen is because as an organism survives often in humans, it doesn't need those genes anymore, right? So they become an open target to accumulate spontaneous mutations that knock them out. Right? If you're hanging out in a chronic infection in the gallbladder, you don't need those guys. Okay? So again, look at this. This is a really important thing because there are so many of those pseudogenes. By the way, we haven't tested them all, but we have tested about 30 of those pseudogenes in an in vivo test. And there are some of the pseudogenes that don't matter, but a good set of them really matter, right? And so this is a way where you can actually take and look at some in vivo, some niche adaptive functions, and be able to test them to determine if they're really true. Make sense? Now let's get away from Salmonella for a minute. Oh, by the way, I should say, we now know that as bacteria become more niche adaptive, We see this in a lot of bacteria, right? As they become restricted to a niche, we see more, even non-salmonella, we see more chromosomal rearrangements and more accumulation of pseudogenes, right? When I was a student, people said, you do not see pseudogenes in bacteria. You don't see pseudogenes in bacteria. And the reason was, they said, bacteria by their very nature have to grow and divide so fast. So if there's a non-functional gene, recombination will remove that gene. That's wrong. It's just wrong. We now know that the amount of energy it takes to maintain a non-functional gene, as long as that's not otherwise harmful, it's not that much energy, right? But in the old days, we say, oh, the ATP required for maintaining a gene in a cell is just going to be a selective disadvantage. Not true. Oh, one other point I might say. Despite all of this great stuff, some of this we learned from genetics. Right? We learned from genomics, rather. We learned this from DNA sequencing. The genomics helped us wonder about certain questions, but it didn't answer the question, right? To answer the question, in addition to having a sequenator, right, you need to have a functionator. And genetics is the best functionator. Because genetic tests demand that you're looking at what's happening in vivo with living organisms. Let's think about antibiotic resistance. And now I'm getting away from typhimurium and typhi, and I'm going to be talking about some work that comes in part from Dharme Hughes' lab, who was typed in the paper.
Can I ask you a question
Yeah.
before you move into different organisms?
Yeah.
So among all the typhi strain sequence in the different regions in the world, are there new pseudogenes or pretty much out of the same pseudogenes?
There are some that are different, but even so, for example, if you compare typhi with other host-restricted strains, some of them you tend to lose more readily.
Okay. Yeah. Thank you.
And I think it's easy biochemically to imagine why that is.
Okay. Thanks.
All right. So these guys wanting to understand low levels of antibiotic concentration, what they do to cells, right? And the issue here is that if you start with a cell, oh, this is salmonella too.
Just trick us.
These guys are geneticists, so what else would they use, right? Okay. So if you start with a situation, let's just draw this like this. All right. And we'll call this ABCD. All right. And let's just say that these loci give you some very low level of resistance to an antibiotic. There are a lot of metabolic pathways that can give you really low levels, so low that if you plate them in the lab, you won't see it. But if you look for growth in tubes with a real strong concentration gradient, you can see a low level of resistance in the very tiny concentrations. Okay? So you treat these guys with a low level of antibiotics, and what you find is they do exactly what we talked about previously with this amplification. What they do is they take those low-level antibiotic-resistant genes and they get recombination events that now give you multiple copies and increase the level of resistance. Just because of the number of copies, right? Now, when this happens and you start to accumulate the number of these genes, and the numbers actually get pretty high, so you can get amplification where you have 200 copies, all right? Let's go back to think about target theory of getting hit with a mutation. Kelly talked some about target theory on Monday or Tuesday, right? The bigger the target, the more likely you are to be hit. So what happens when you've amplified the target? Right? What that means is the probability of getting a mutation in one of these sites that increases antibiotic resistance is high, right? Maybe 200 times higher than it would be otherwise. Right? And so what this does is the low level of antibiotic resistance can allow these processes that select for high levels of antibiotic resistance. And once you have the high level of resistance, if you're still exposed to antibiotics, you can do the opposite and collapse to a situation where you only have the high level of antibiotic resistance. So this process goes back and forth. As long as you're selecting for antibiotic resistance, you will always maintain that resistance. Okay? All right. Now, if you think about what's happening in these cases, it was found often if you take the bacteria that have low level resistance, they may have 2 copies of a particular gene. And as you expose them to higher levels, those copy numbers go up, and then you see this collapse down to a single copy afterwards. Everyone knows why low-level antibiotic resistance is so important? Yeah? Why?
It keeps the resistant strains over there. Is that what you ask? Having low concentration of antibiotics?
Yeah. So the low level of antibiotics, as confirmed in these studies, can result in high level of antibiotic resistance. Okay? And here's the basic issue. This is tied to the thing of having your idea of a complete dose of antibiotics, or taking antibiotics when you don't really need antibiotics, because then you tend to have these low levels, right? The infectious disease docs that I know now in the United States say, we're going to give a high level of antibiotics for 3 days. If it's not working, that antibiotic isn't the right one, right? And then boom, you're going to go off that high level, and it either cured you or you need something different. But they don't set up the situation where you have an antibiotic dose that goes over 2 weeks, and potentially you start decreasing it at the end. All right. Okay, so now we have antibiotic resistance. Here's the problem. Antibiotic resistance often results in a fitness cost. Right? So, if you take and you want to plot growth versus time, and you look at that in a situation where you have antibiotics, of course, the antibiotic resistant mutant will grow nicely, and the antibiotic sensitive mutant won't grow. Right? There's nothing magic about that. But look at what happens if you take these guys and you plot them the same curve, and now you're doing this without antibiotic. What often happens now is that the antibiotic sensitive strain grows well, and the antibiotic resistant strain grows less well. So this is what we'll call a fitness cost. This is in the absence of any antibiotic, right? It's still influencing that cell. Now the problem when you have a fitness cost is that, let me back up a second. This is really important what I'm talking about. Antibiotic resistance, when it accumulated early on, people said, oh, if a hospital has a high level of resistance to ampicillin, we'll just tell the physicians in that hospital to quit using ampicillin for 6 months, use some other antibiotic. 6 months later, because of this fitness effect, 6 months later, the resistant ones will have disappeared, right? And we can shift back to ampicillin, right? So it influenced the way hospitals used antibiotics around the world because of this belief that this fitness cost was so critical to survival of the bacteria. Right? So it went beyond science to policy. Here's the underlying problem. When you take a look at this kind of situation, what you find is that you can accumulate mutations, compensatory mutations, and now if we repeat this curve, what we see is the antibiotic sensitive and the antibiotic resistant go the same. Point mutations in the chromosome. So far, only a limited number of these compensatory mutations have been mapped. In most cases, they're in metabolic genes. So what this means is that this concept of fitness cost, rather than providing a way for us to respond to antibiotics, it results in these particular mutations that now have high-level antibiotic resistance that's just going to stick around. They're just as fit as the wild type, right? So by understanding those mutations, we may be able to figure out new ways of using multiple antibiotics or multiple approaches that will influence that. At this point in time, it's really an open question.
Just make one comment. So those Swedish guys actually managed to talk every physician in the country of Sweden to not use trimethoprim for urinary tract infections for 6 months. And then checked after 6 months, nothing changed. The resistance was just the same. The whole country. Amazing.
Yeah, it is really amazing. That Sweden is a very law-abiding country, right? And so you can get that. It's really an interesting thing, but it's a psychology in the country. You can get that out. All right. The last thing here that I think is really important in thinking about is genetics and evolution and how they come together in ways that matter to us is that very often when you see evolutionary processes in bacteria, it's from acquisition of a new gene. And for a long time, these new genes looked like they came from Mars. I mean, we couldn't identify them, right? But now that we can do metagenomics, what we're learning is that out in the environment, there are lots and lots of these new genes that can take and convert a toxin non-producing strain to a toxin-producing strain, right? And we find these kinds of gene pools even in what looks like the most pristine of environments out in nature. So we mentioned something about gene exchange between bacteria before. It's happening all the time out in nature at high levels, right? And we see it when we select for it as humans. All right. The goal today was to just take you away a little bit from thinking about just constructing your favorite mutant to understand your favorite virulence gene, to think a little bit about what's the big picture, what's happening all around us, right? And how can those genetic approaches allow us to answer these big problems, right? Okay, questions? Yes.
During the duplications, you also can generate these pseudogenes, right?
Oh, yeah. So sometimes a duplication of a gene, right? So now you have 2 copies, and you accumulate a mutation in one of those copies, but the duplication collapses, and you're left with the pseudogene. Right? So now, if there's a benefit to having the wild type gene, you almost always will lose the copy with the pseudogene. right? But if the pseudogene doesn't matter under that particular growth condition, it's a 50-50 proposition.
Why to keep a pseudogene? I mean, eventually it's going to disappear.
Eventually it will disappear, right? Just because of random deletions. But if it doesn't have a negative selective phenotype, it just hangs around. We all have friends like that.
About chromosomal rearrangements. So, what about those bacteria that have more than one chromosome? I mean, that's because salmonella is only one big one.
Yes, yes. And actually, one of the reasons I got really interested in this initially was from bacteria that have more than one chromosome and thinking about that. Bacteria that have more than one chromosome, some of them seem to have two stable chromosomes. And they don't really share a lot in common, so they don't have the homologies to allow it. But many bacteria with multiple chromosomes actually undergo recombination, so it's back and forth. Borrelia does this very high frequency. Vibrio does this.
Yeah.
I mean, it's pretty common to have a lot of that gene exchange. We started thinking, we drew our chromosome, we had 0 and 100, and this is here and this is here. We talk this way, and we have strains that allow us to do that, where we can do really fine structured genetics. It's really good. But in nature, there's a lot of weird stuff happening because it can, right? A chromosome, bacterial sex is kinky. Right? Okay. So the paper today that I talked to you about is from Dr. Hughes' lab, who we mentioned already. And basically, the things I wanted you to think about from that paper are the impacts of going through a bottleneck, what that means, right? And we designed some genetic approaches to look at what's happening in nature. But what they did was they said, look, if we want to have a bottleneck, what they do is they take and they grow bacteria multiple generations in liquid cultures, so they all have a chance. But then they take them out and streak them on a plate and take a single colony, right? That's a bottleneck, right? So it allows you, if you do this with multiple cultures, it allows you to understand what's happening with that bottleneck, right? In a very controlled way. So what they've done, we were taking strains from nature to look at this, and in this paper, they set up a scenario in the lab where you could look at it. It's a really beautiful set of experiments. So that was one of the key things I wanted you to think about that. And the other thing, actually, that's interesting in their paper, one of the key things was they were looking at duplications. Does anyone know how they selected for this? Right, so salmonella can grow at very low levels on malate. It doesn't grow very well. The reason it doesn't grow very well is malate's not transported in the cell very fast. Right? They grow faster on other dicarboxylics that are transported faster. But because it doesn't grow very fast on this, if you increase the number of copies of the transporter, then they'll start growing faster, right? So you have your genetic selection. It ends up that in bacteria, often the rate limiting step for growth is transport of a certain substrate into the cell, right? It's not a problem for glucose, but it's a problem for a lot of other kinds of substrates organisms encounter in nature, right? So the idea here was to simply look at how you can set up experiments in the lab that will allow you to fix a particular genetic trait to reflect evolution in process.
So your screening is from tiny colonies to larger?
So the initial selection that they did was in liquid media, looking for guys that grew faster, right? And then they transferred them and transferred them and transferred them. And as they transferred them, they're going faster and faster. Then once they had something that grew fast, they streaked it out so they could take individual single colonies. And if there's multiple mutations, you'll be able to separate them out. You can do a lot of really cool stuff with bacteria, from understanding at the molecular level how a flagellum works, right? A really interesting and important question, to being able to understand how microbes evolve in vitro. But to do it well, genetics really provides tools to confirm your hypotheses. That's a take-home point. And if you work on insects, there's a lot of cool stuff you can do in insects. All right. That's all, folks. You have any questions for Kelly? Let me say one other thing, and that is Kelly up here said, if you want to do it, tell me what you want to do. But he's around for another day or so, and if you're really thinking about something you'd really like to do, nab him, make him sit down with you in a corner and draw it out on paper.
Bring me a beer.
Yeah, that would work. Hey Kelly, why don't you tell them what you mentioned to me? Why not using CRISPR on bacteria?
Well, CRISPR makes second site mutations for one thing, right? So you can do a CRISPR targeted, but then you've got other stuff going on. And that doesn't happen with lambda red.
Yeah.
That's one example.
Yeah, okay.
And the other example is that, let's see if I can get this right. So if you want to target a particular site, whatever your favorite gene, you need to express this Cas9 protein, right? They need to express this guide RNA, which can then target the cutting, right? And which can cut other places. But then you also, in that situation, this has to be constitutive, and then you turn on the guide RNA, and that can be P arabinose or whatever you want. And then to make it actually really work, you have to electroporate your DNA. So you're going to make your cuts, Cas9 cuts, right? But now you have to have DNA that you're going to replace that with. And so you also want to turn on lambda red to make this efficient.
Yeah.
So it's really a very complicated scheme. And we're playing with this, and we find just the plasma we got that they have in E. coli, it makes salmonella grow like shit. Really. So now we have to find a new plasma to make that. Yeah.
But along those lines, you always want to backcross. You always
Yeah,
want to
so once this is done, you would want to move it back into a clean background because of all the other hits in the chromosome that Cas9 just does.
Okay. Yeah, that's also a danger for using higher organisms, to call them some way.
Bigger organisms,
Bigger.
I call
Yeah, yeah, anyway, yeah, that's better.
them. Yeah, no, they have these second site hits. It's a real problem. I mean, they're trying to improve on that, right? That's obviously the smart thing to do is to work on that. It also has, a disadvantage, if you know about this CRISPR-Cas9 system, is that these things, the traditional one actually requires nearby where you're targeting this, what's called a PAM site. So a GG right before, and if you don't have, that's every 16, but sometimes you just don't have one. So people are trying to find PAM sequences that are different than GG, so they can target more areas. But if you don't have a GG in the area, too bad.
Yeah. Preguntas?
Yeah.
Did you know if the method of lambda-ret could work in mitochondrial chromosome?
That's a eukaryotic system. And
Mitochondria is a bacteria.
yeah, but in a eukaryotic cell. The correct answer is I don't know, which was your question, so I'm done. I don't think it would, because you'd have to get it into the mitochondria. I don't know how you do that. But you need to talk to a person who works on mitochondria. Can you grow mitochondria outside cells? No.
No.
Can you get DNA into yeast mitochondria? Maybe. But I don't know. Good question.
But just somebody in here earlier today told me they work on insects. This opened the door for doing things in insects that were impossible before, right?
Absolutely.
More questions? Andrea.
From rearrangement and with the genes can be the causes of the opportunistic diseases, or it's another mechanism for that?
So it could be the cause. I mean, it could be a virulence factor, right? I don't know of any that is a virulence factor. I know of cases where I can say theoretically that would happen, but I don't know of any. But that's just the amount of knowledge in here.
More questions? What about we do a little practice for the song? So we can have the graduation tomorrow. Maybe, I'm not sure, Marta is the one who's going to direct it. So she's the one who wants us to do it better than the Chileans.
Yeah.
No.
Come on. At least maybe we can get the tune. Then we can get we need first to get the tune and then the steps.
Actually,
You need to move here. So the video.
Por favor.
Si quieres grabar, ponle. Quítale el cable y empieza a grabar. Por favor. Si quieres, quítale el cable para que sea más fácil.
You're going to cause an international conflict between Chile and Mexico.
No, we're good friends. Ándale, Marta.
And then we'll translate it into Spanish and do it again. Yes.
Can you just give us the tune? I'm a little teapot. I got cloud.
Two promoters both point out.
Both point out.
Do it like a rhythm.
Silently hear me shout. I turn genes on all about.
Again. Sing together.
Teapot, I got cloud. Two promoters both point out. Feeling tetracycline, hear me shout. I turn genes on all about.
Last one.
you want to see a competition?
No.
Turn the sound.
De Chile, ¿cómo se dice?
It's nice to know your competition.
I'm a little teapot.
Pero creo que nos faltan los pasos.
Ah, sí, excelente. Pero acá afuera. Y un huevo. Hay que hacerlo acá afuera parados. Ahí donde están implantando los honorarios. Hay que echarnos adentro de los honorarios.
That's for the dancing. Come on. She's your turn.
¿Cómo era la coreo? A ver, Amanda, tú te sabes la coreo. Es que era Amaleo.
Uno, bueno, ustedes bailenle, yo nomás intento cantar.
Amaleo. A ver, otra vez. Amanda.
Two promoters both point out. Imitate the cycling, hear me shout. I turn hints on all about.
Otra vez. Otra vez. Pero todo. Sí, creo que sí. Pero es que
Hagámoslo.
Ok. Come on, Kelly, come.
Kelly, please.
Please, help us.
You need to lead us.
Help us, please.
Come on.
Shout us. Come on.
Here we go again.
We're slow learners. Come on.
Okay. Someone say 1, 2, 3.
Una, dos, tres.
I'm a little teapot, I got clout. Two promoters both point out. Feed me tetracycline, hear me shout. I got this song all about. Bravo!